to this meeting is being recorded. Thank you so much, everybody. Thank you, everybody, for your attendance. Thank you to Bob, to Dr. Bob Fosbury. This is going to be a really interesting conversation, I think, presentation and conversation. Honorary Professor at UCL Institute of Ophthalmology, and you will get that one wrong.
I don't really know what it is, actually, but you know, it's a good home.
Thank you for being so graceful. But yeah, I think I was practicing it, Bob, while we were waiting. Specializing in the interaction of light with biological systems. Previously, a senior European Space Agency astronomer for the Hubble Space Telescope project, Professor Robert has shifted interdisciplinary research on natural coloration and the effects of near-infrared light on human health. Thank you for this. Thank you to Meg for setting it up. It's something that we've been discussing in the channels quite a bit, and it's truly fascinating, and I think will change many of us by the end of the talk. Thank you. Over to you, Professor Robert.
Okay, I'll share my screen and find where we are. Sorry, I'm going to start again.
Take your time while I practice that ophthalmology word.
Sorry, it's taking a while. Does that look
Yeah,
vaguely right?
Editing the Sun, great title, by the way, Bob.
Yeah.
Great title.
Right. Okay. Well, let me start. This is quite a long story. We've been studying it for quite a long time now, and it's one of those stories which has started because we've missed something. We made a mistake maybe 10 or 20 years ago, and now we're just discovering the consequences of that mistake, and we're trying to understand what it is. So I'm going to start off with what I call a visual abstract, which contains the essence of the talk, and that's that we're really realizing that life thrives. I call it starlight because I think this is quite general for life on any planet. But we thrive on the sunlight that casts its shadow. So if we're standing out in sunlight, we're casting a shadow. The light that would have filled in that shadow is going into us in some sense, and it's having what we think is a profound effect on our biology. So that's the story in a nutshell. But just to give a brief overview and introduction, I'll introduce the topic I'm going to be talking about first of all, and then I'll have a short interlude where I'll introduce some of the clinical data and the experimental data that we're working on. That will not be the focus of my talk, but it's the data on which we base the work that we're doing to try and understand what's going on. So this is all fairly obvious, but life on Earth has evolved under sunlight for more than 3 billion years now. We're now realizing that the origins of life go back very far further than we perhaps thought before. And over less than a century, we've moved indoors and changed the light bulbs for reasons of energy efficiency. We thought, oh, great, we can get new light bulbs, produce lots of visible light, cost much less in electricity, hunky-dory. But what if that change was not biologically irrelevant? So this talk looks beyond photosynthesis, which general we understand reasonably well, and vision, which we also understand reasonably well, I say reasonably, to a deeper and older level of bioenergetics. It's really a story about water, mitochondria, and the molecular machinery that keeps us alive, keeps us going. And I'll try and use analogies to make some of the unfamiliar physics a little bit more broadly accessible. And the analogies are quite close, actually. So it's quite a good way of explaining some of the things we're talking about. And I'd like to stress that, the ideas I'm thinking about are not a finished theory at all. But I do argue that pursuing it now is going to be very important, and we may have to undo some of the edits we've made to the light we're living under indoors. So the clinical and medical evidence I'll come to first of all. And I say it's not the focus of this lecture, so I won't spend long on it. We know very well now from a wide range of experiments and a wide range of clinical results that mitochondrial respiration increases under red and infrared illumination. We can stimulate our mitochondria to work better when they see red and infrared illumination. There's lots of experiments. It's called photobiomodulation about accelerated wound healing and tissue repair. This is really quite dramatic, and I've used it myself on a number of occasions now. I may explain a bit later, but it's quite dramatic, and especially when you use the right kind of lights. We know it helps with neuroprotection and functional improvement. These are experiments done, by the way, mostly by us on humans now. We do all these things in vivo. There are a lot of earlier experiments that were done by Glenn Jeffrey and his collaborators in London on fruit flies and bees and mice over the years. But the fly experiments have been very informative. And we know that the results we're talking about are conserved across this very wide range of species. So we're not talking just about humans. We're talking about life in general. In fact, essentially the entire biosphere. One of the results we get from the short-lived animals is aging and stress resistance. We can increase the healthy lifestyle of flies and bees and mice very readily by using light in an intelligent way. So across many, many systems, long wavelength light improves biological performance, where control, reorganization, and resilience matter more than raw energy supply. Now, what I'm saying there is that, we kind of understand photosynthesis, and photosynthesis is taking photons of light from the sun and converting that photon energy to chemical energy really quite directly. So you're taking photon energy and you're feeding it through this photosynthetic process, the two systems and so on, and you're ending up with chemical energy at reasonably high efficiency, not super high, but reasonably high efficiency. What we think is happening with what I'm going to call photometabolism, which is the partner of photosynthesis, is that it's not supplying energy to us. It's not supplying energy as food. So we're not metabolizing energy with the process we're talking about. It's a much more subtle process, and I call it a process of lubrication. It lubricates the machinery of respiration in a really rather subtle way, which I'll come to. So in that sense, it has to be distinguished from photosynthesis. It really is a partner to photosynthesis because we're both using sunlight for both of these processes, and they're both working through the mitochondria. Now, there's a wide range of clinical literature as well, and we're collaborating very closely with an American medic called Roger Schwelt. He's well represented on videos in the US. He's a medical educator. He runs a company called MedCram for educating doctors. So he has many videos on his video channels, and a lot of them are about light. And he made very significant leaps in understanding about the effects of light on biology during the COVID period, which is when I was following him. I subsequently got in touch with him, and now we're collaborating very closely. So the clinical literature tells us something very real is happening. What it does not yet tell us clearly is why. And that's the kind of gap that I want to try and fill in today. I think a lot of this work has been done very recently. We're just beginning to understand it, but I think it's probably an important process. So this cartoon here represents really what we know about metabolism, and we have the sun sitting up there. I'm going to be talking about spectra in terms of energy generally rather than wavelength for reasons of biological clarity. So the peak spectrum of the sun appears at 0.75 electron volts, which is about 1.6 microns. It's a different representation of sunlight that we use in everyday life of normally watts per square meter per nanometer. We use different units of photon flux per square meter per electron volt, per unit energy, that is. But the sun peaks at about 1.6 microns and in terms of photon flux. But the photosynthesis is done at much shorter wavelengths, between about 1.8 and 3 electron volts, where we know that it converts these photon energies, visible photon energies, into carbohydrates, into sugars. And those carbohydrates are what we either eat directly or we eat through the chain of predators and so on. We eat as meat and so on. So the energy from the sunlight is going into the plants. It's being transferred from the plants into living animals, and it ends up in the mitochondrion, where it ends up converting converting the food into adenosine triphosphate, ATP, the energy currency of the cell. So this has been known for a long time now, and it's been the process of photosynthesis has been studied in great detail, particularly in the early 2000s, where high-speed laser physics experiments were done, and the way that photon energy is transferred into the photosystems was really rather well understood at that point. So what we're realizing is we're missing a channel here. There's another channel from the sun which goes directly into the mitochondrion by penetrating the body. And this is the infrared light, the longer wavelength light. Visible light can't get into the body because you have these very strong pigments in the body which will absorb it very close to the skin. But in the infrared, the situation is quite different, and the light can get deeply into the body. And what we realize, for healthy metabolism, you need both of these channels open. And if you don't have them both open, your mitochondria age more quickly, they become less efficient, and you don't live as long, basically. It's all to do with the rate of aging. So for healthy metabolism, we need a broader spectrum of light than just the visible spectrum. Of course, life doesn't always see sunlight directly. It can be reflected, scattered, or filtered. Obvious example is the sky, where certainly in Britain at the moment, we don't see the sun directly. I've hardly seen the sun since the new year. But so clouds scatter and mix direct sunlight with blue sky together. So the result is daylight. And without direct sunlight, daylight is weaker than direct sunlight, but it's bluer than the light reaching you from the sun, because it's added the blue from the sky back into the solar spectrum. I do solar spectroscopy all the time, so I understand this stuff because I do it every day. That's relatively straightforward. What we realize, the forest understory is tremendously important. It's a very special case because it's so relatively rich in near infrared. When you walk into a rainforest or into any deeply canopied forest, you get many times more photons in the infrared than you get in the visible. And we think this is one of the reasons why forest environments are so biodiverse, because it's a tremendously good place to live, because you're not subject to direct sunlight so much, which can be damaging to you under some circumstances, but you do get a very rich source of infrared. The other interesting story is underwater environments, which are blue. They're very blue rich, but they're very infrared poor because the infrared is absorbed very quickly in bodies of water. So one of the questions, which I'm sure somebody is going to ask at some point, is what about the animals that live underwater? I won't try and answer that now, but it can be discussed. I want to give a sort of review of natural daylight, first of all, because I mean, as an astronomer, I've grown up thinking about starlight, and I also, as a hobby, studied sunlight and daylight for ever since I was a kid. I'm very fascinated by the nature of natural daylight, and it's very varied. So this diagram here, we produced this for an ecological journal a year or so ago, and the first thing it shows, if you look at the scale on the left here, the y-axis scale, I seem to have, sorry, I thought my cursor was showing, but it's not. Anyway, look at the y-axis there, irradiance in the visible spectrum. That covers 10 orders of magnitude. So actually it covers 9 orders of magnitude. It's got a little bit spare at either end. So our visual system can cope with 9 orders of magnitude in dynamic range, which is extraordinary. And as the sun sets and we go into twilight, we've gone down in brightness by a factor of a few thousands. But when we go down in twilight to a really dark sky in a dark environment with no moon, we go down another 7 orders of magnitude and so on in brightness. So the contributors to this natural light, of course, is sunlight directly, scattered sunlight, scattered from the clouds, scattered from your surroundings, scattered especially from vegetation. Vegetation scatters infrared light extremely efficiently, which is why when you see infrared pictures, all the leaves look very bright. The moon, air glow and aurorae, and of course stars. When we get down to the bottom of this diagram, astronomical twilight, we get a significant contribution of the mass of starlight in the universe to the light that is available to us. Now, I've spent many months of my life on tops of very dark mountains in Chile and Australia and Hawaii and places. And I know that if I'm outside on a completely dark night with no moon, I can see very, very clearly. I can see around me. There's still plenty of light to see. I can't see in color, but I can certainly see light clearly. So it's important to remember that daylight does have this enormous dynamic range. And a lot of the processes we're thinking about are in some sense logarithmic in action. So we're often talking about ratios rather than absolute quantities, and I think that's an important point to take note of. This is one of my pictures of the infrared environment. This is taken at about 800 nanometers with an old-fashioned Sony infrared camera, which I bought in about 1999, which had the Attenborough mode, where you could switch the infrared filter off the detector and you could take infrared images for seeing animals at night. They stopped manufacturing those when they realized that bathing suits were completely transparent in the infrared and they didn't want to get sued. So that was unfortunate. So it's harder to get infrared cameras now. But you see at 800 nanometers, the vegetation is incredibly bright. It can be brighter than white clouds in the sky. And that's because vegetation needs to get rid of light that it's not using for photosynthesis. Plants have mitochondria, they have to metabolize as well, but the light they're using for photosynthesis is in the visible. And they don't want to have their leaves heated unnecessarily by infrared light. So they've generated very, very effective ways of rejecting the infrared light, which you can see in this diagram very obviously. And that means that if you're in the forest understory, you're getting a rain of infrared radiation from the canopy, which is, we think, extremely healthy. So the healthiest place you can sit in the world, I think, is under the shade of a tree in the summer, surrounded by blue sky, when you're actually has the sun shaded from you by the tree, but you can still see the blue sky. I'll come to that at the very end, and that's a very obvious situation we can be in, but that's a very, very healthy place to be in because you get the richness of the infrared source with you. I was talking about the forest understory. Unfortunately, I don't have a rainforest in my garden, but I do have a forest of ferns. So there's a tree fern there, and there are a number of other normal ferns underneath. And if I stick my spectrometer under all that in the deep shade, under all those ferns, I get a spectrum there. You see the intensity and the wavelength on the bottom. You can see the thin green part below 650 nanometers is the visible spectrum, which is very much diminished by all the vegetation above it. But as soon as you get to 700 nanometers, you get this enormous flux of infrared radiation, which is rejected by the leaves, by the ferns. And so it's a very, very rich infrared environment, and we think that's metabolically extremely beneficial. Now, many people think that it's very healthy to go into forests because of terpenes and so on. Actually, I think it's mostly because you're getting a huge flux of infrared radiation without getting some of the damaging shorter wavelengths. So the understory is a very special environment for metabolism. Now, just to illustrate some of the things that contribute to natural light, lovely picture here taken in Latvia of the aurora and the Milky Way at night. So you can see the mix of some of the things that we were discussing in my plot before. This is our observatory in Paranal in the northern Atacama Desert, the very large telescope. These skies are very, very clear, and you can see this long, thin triangle sloping up from the bottom right to the top left, which is called the zodiacal light, and that's just scattering of sunlight from dust grains left over from the protoplanetary disk that formed the planets. So there's still a lot of dust in the plane of the solar system, and that acts as an efficient scatterer of sunlight. So it's only in very dark sites that you can see that clearly. But the most interesting one is this one taken. This is the very large telescope is on that mountain peak right in the center of the picture. And we can see the Milky Way. It's a wide panorama, so the Milky Way is curved. It's not really curved like that, but because of the panorama. And you can see there are very few sources of artificial lighting there. There's a bit of lighting at the observatory, very low level. There's a bit of lighting at the distance, which is near the coast in Chile, just south of Antofagasta. But you see that lovely red and green glow. That's the air glow. And the air glow is particularly strong in Chile because of the South Atlantic anomaly. It's a magnetic anomaly. And so we see this quite strongly. You can't see that in color with your naked eye, but just a simple digital camera will do it for you with relatively short exposures. And that's very interesting because the oxygen which is producing that air glow is the result of photosynthesis. And so we wouldn't have that oxygen free in the atmosphere without photosynthesis. So it's a very obvious, very strong biomarker. Anyone who saw that from the outside would know that there was something other than chemistry producing oxygen in the atmosphere. So, this is a biomarker which we'll obviously consider in looking at other planets as well. But it's a really rather beautiful illustration of the range of different mechanisms and colors. The red and the green here are the same mechanism that you see in the aurora, but they're excited in different ways. This is excited by sunlight during the day into long-lasting metastable states in the oxygen atom, which can release it slowly at night. In the aurora, of course, you're using high energy particles from the sun to excite the spectra. So what I'm going to show you is the spectrum now, the solar spectrum and the spectrum of some lamps. But I mentioned that I was using a different form for expressing these spectra. And I put this here just for reference. It doesn't matter terribly much here, but if anyone's interested in knowing how to do these transformations, it's worth looking at this. And the solar irradiance spectrum is a histogram, of course. It's not a XY point plot, it's a histogram, it's binned. And so when you transform it into other units, you have to take account of the bin width. And so it's a slightly more challenging transformation than you might imagine, but it's not difficult, but it's slightly non-trivial. So for bioenergetics, we're interested in the number of photons that the biology sees and the energy of the photons. So we choose to use this photon flux, which is the photons per second per meter squared per electron volt. And the spectrum does look quite different. And you see in that equation, you have a lambda cubed. It shifts the whole spectrum into the infrared, which looks weird, but it's what the solar panel manufacturers use because they're interested in getting electrons from sunlight in the same way that we are in biology. And so they use the same kind of transformation. And this is what it looks like. The red is on the left, the infrared is on the left, and the short wavelengths, the blue and ultraviolet, are on the right here. And just to orient you, I've shown two very common sorts of lamps here. The green shaded area on the right is a typical white light emitting diode that emits just in the visible spectrum. And we all know these diodes are powered by a blue LED, so they have extra blue emission, which shows that's the peak on the right, the little extra peak on the right of this green shaded area. But of course, these are designed to only produce visible light. And so if you look, there's a slightly darker redder line at the bottom there, which is actually a measurement of a tungsten filament lamp. And it fits a black body extremely well. And I've extended it as a black body all the way into the infrared here. So you can see the kind of spectrum which is emitted by a tungsten filament, an old-fashioned tungsten filament or quartz halogen bulb, is very, very different from the energy that you're getting from a white LED. And your people who are saying, why aren't we sick in the indoors? If they really spend all their time indoors, they will get sick. And it won't happen in a day or two. It'll happen in a longer time. But so, and on this diagram, I've also got the spectrum of the sun. This is the spectrum that's reaching the ground level in a clear sky, and you can see all these deep absorption lines on the left-hand end here, which are mostly water. So water is a strong absorber. And in the visible spectrum, there are very few absorbers. So the air is very transparent in the visible part of the spectrum. There are weak water lines, there are weak other lines, but basically there's very little absorption in the visible spectrum. But as you move into the infrared, the absorption becomes much stronger. And this is very important for us. So there's this huge red area in this, it is a logarithmic plot, by the way, so I'm cheating slightly. There's this huge red area on the left of this diagram, which is near infrared and infrared light, which we're not getting from white LEDs. And neither are we getting it from modern low-E glasses, which reject the infrared in order to avoid thermal imbalance in buildings. You don't want too much heat going into buildings in many places. So that's what the spectrum looks like in my new units. And you'll see the peak here is at about 0.75 electron volts. And actually it sits a little bit above the Planck function here. The red dash line is the thermal black body spectrum computed using Planck's formula, which would emit exactly the same energy as an object with the same surface area as the sun. It's called the solar effective temperature, which is about 5,772 kelvins. And so the Planck function, you can see, is very close to the continuum of the solar spectrum, but the solar spectrum peaks a little bit above it, about 0.75 electron volts, which I'll explain in a moment. That's quite significant, in fact. So I've divided this region into three spectral regions. Region 1 on the right, which is the visible and ultraviolet spectrum. Region 2 in the middle, which is the region where the body is relatively translucent. This is where light can get into the body. And those blue vertical bands in this diagram represent where light can get through these water absorption bands into the body. So that's where we think a lot of the effect of the light, the infrared light, is carried out right deep in the body, not just on the skin. And then as we move further to the left, water becomes more and more strongly absorbing, and most of the sunlight is absorbed in the skin or very close to the skin, rather like in the ultraviolet. In fact, we think those two processes are coupled. The damage that's done in the ultraviolet, we think, is repaired by the infrared penetrating to similar depths at much longer wavelengths. So I call region two the cathedral effect, which I will explain in a moment. That's one of my analogies, but let me just move on in this diagram. So what we've done over the last 20 years is we've wiped out the entire left-hand side of that diagram within the built environment. So we only have white LEDs now, and in general, and the bigger buildings, not domestic buildings in the UK yet, but large buildings, you know, the big skyscrapers in London and so on, they reject the infrared. So if you're living in these skyscrapers, you're completely infrared starved. And just as an example, if you just look up the rate of type 2 diabetes in Saudi Arabia, the rich people, not the immigrants, the rich people who are living in these big skyscrapers, 36% of people suffer type 2 diabetes. It's a huge effect. So what we've done is we've basically wiped that out. We're starving ourselves of infrared light. And I coined this term 21st century scurvy. Its symptoms are not dissimilar to scurvy, and it develops on a timescale which is very similar to the naval people who sailed down to the Cape Horn and Cape of Good Hope to sail round. They most of them had scurvy by the time they got there because it took them several months to get down there. So we think this scurvy is being suffered on the space station at the moment, and probably in other environments like submarines as well, where people are starved of infrared light. They've taken all the tungsten filaments out of submarines and the space station now, and they rely on infrared lights. Although they use so-called circadian lighting, they change the color temperature of the lighting throughout the day and night, their day and night, artificial day and night. That doesn't supply any infrared light, so they still get sick. The circadian rhythm lighting doesn't do terribly much for them, I don't think. So how does light interact with living tissue? Now, this is really the crux of the problem. Light penetrates into living tissue in a way that depends critically on its color or wavelength. For UV invisible light, tissue contains these very strong pigments, or chromophores we call them, that result in light being absorbed very close to the surface. So we can't get light deep into our body in the visible. And all the effects of light absorption are occurring in the superficial layers. In the blue, it penetrates slightly deeper than the skin into the fatty layers below the skin. And I think that's now understood to be the place where most of the skin cancer is developed. It's not developed in the skin itself, it's developed in the underlying layers. But there are people who are much more expert in this than I am. So in the near infrared, that is, from beyond the visible spectrum that we can see through to about 1.5 microns in wavelength, the chromophores are extremely weak. And the photons, if photon gets into my hand in these wavelengths, it's very unlikely to get absorbed by the first thing it hits. And it will scatter off that. It could be a cell boundary or something. So the infrared photons get into my hand, into my body, and it will scatter many, many times before it hits an absorber that's strong enough to absorb it. And so what this means is that the light is being absorbed, not being absorbed, it's penetrating your body, and it's basically scattering around. And I call this the cathedral effect. If you walk into a cathedral, an empty cathedral, and you clap your hands, you'll hear this echo going round off all the walls for ages afterwards. Makes it terrible to play high-speed, detailed music if you're playing an orchestra or something because of the long reverberation times. So effectively, we have a long reverberation time in our body in the infrared, and the light will scatter maybe tens or more times before it hits an absorber, which is strong enough to absorb it. But for all the light that gets into the body, it's once it's got into the body, it's very unlikely to get out again through a surface. And so it will be absorbed. However weak the absorber is, it will be absorbed in the end. So basically, about half the light that hits human skin, for a light-skinned person anyway, about half that light penetrates into the body, and very little gets out again through the skin. So basically, you're getting something roughly like half the light in the infrared is getting into your body and it's bouncing around many times and it's being absorbed by chromophores. So further into the infrared, the water absorption, as I said, increases in strength, again limiting the depth of penetration. But we think that's doing different things from the light getting deep into the body. And this illustrates what's happening. I mean, I photographed my hand about 2 years ago now, and it's been shown so many times on the web around the world that I think I probably have the most famous hands in the universe now. It's even in a Nature paper now. So I used 850 nanometer LED light into shielded, getting into my hands to take those pictures on the left. And you don't see the bones at all. You see the veins because deoxygenated hemoglobin has a significant, not very strong, but significant absorber at around 850 nanometers, about seven, it's about, I can't remember exactly the wavelength, but it's near 800 nanometers. And so that shows the veins in shadow. And of course, the veins are much closer to your skin on the back of your hand than they are on the palm of your hand. So you can only see the veins relatively weakly on my palm. On the right, there's an experiment done by my colleagues in London with shining sunlight, in this case, through a human body, very carefully shielded from stray light, where you can see the light gets right into your back, right through and comes out through your chest. These are all the hairs on your chest. And you can see the veins beneath the skin. You can see these shadows of faint veins underneath the skin. So the light really is getting into the body. And a tiny, tiny fraction of this is getting out. This is a very difficult photograph to take. Most of the light gets absorbed in your body, and a tiny, tiny fraction of it comes out again. And again, another spectrum. I apologize for so many spectra, but we are talking about light of different wavelengths, so I can't really avoid it. The black line, again, is the solar spectrum, but here I'm talking about reflectance and transmission, not photon flux or anything. So it's a different y-axis. It looks very similar, so don't worry about it at the moment, but it's basically the transmission. So the black line is the transmission of the solar spectrum with a solar altitude of about 45 degrees. So you can see the scattering is decreasing the visible light to make the blue sky, but the water is the dominant absorber in the infrared. Now, the red line there is the reflectance of my hand. And what I do is I shine a tungsten filament. It's actually an integrating sphere on my spectrometer, which has a small port on it, which emits tungsten filament light quite powerfully, bright as sunlight, onto my hand. And I look at the light, which is, it's not reflected from the skin so much, but it's backscattered from the scattering process. Excuse me, in my hand. So about 50% of the light at around 800 nanometers here is leaving my hand, but the rest is going in. Now the blue light there is the light that's coming out the other side. I can do the same thing. I didn't do this with my chest. I did this with my hand. My hand is about 3 centimeters deep. So I can see light coming out of the other side of my hand. And so the main transmission peaks at about 1.5 electron volts there, which is about 850 nanometers in wavelength. And you get some coming out in the infrared as well. My spectrometer is not sensitive enough to measure it into the infrared. Now, this is very, very weak. I've normalized it to 100% here, but there's actually many factors of 10 below the reflectance level. It's just to show that you can get light through the body and what the absorption spectrum actually looks like. So that's what happens. And we also have to remember that clothes also become transparent in the infrared. I mentioned the bathing suits, but normal clothing, this is just a woolen jumper, a shirt, and a t-shirt hung up on a sort of 6 layers of clothing there in visible light. I'm shining a red light at 660 nanometers through that clothing, and you can, depending on how bright your screen is, you may see a faint glow coming through the middle, but basically that's blocked essentially completely by the clothing. And at 850 nanometers, I get a very clear signal through the clothing. So in order to get the benefits of infrared light, you don't have to strip off. With normal everyday clothing, you have your whole body which can absorb infrared. And that's very important to remember. It's not just the exposed parts of your head and your arms and so on. You're getting infrared getting in, and your clothes are obviously protecting you from the UV. But so, and just to look at the nature of this scattering, there's a nice little picture I took of my finger, again over the port of my integrating sphere. So there's a bright white light under there, and you see it glows red. I think most people think that if you do that, shine a flashlight through your fingertip, the light coming out is red because your blood is red. But blood has very little to do with it. I've got a bamboo spoon here, which has a thickness of about 2 millimeters, and I put that over the port of my integrating sphere. There's no red pigment in there at all, but it's come out red. And that's simply the setting sun. You scatter the blue light first, and you scatter the red light last. So the penetrating light is the red and the infrared light. That gets much deeper into living tissue than anything else. So it's a very graphic demonstration of light produced by multiple scattering process rather than pigments or dispersion or anything. So it's a very important scattering process. And this is what gives you these beautiful twilight colors when the sun's gone down below the horizon. You get this lovely twilight from deep purple blue above you down through the blues and the apple greens, the oranges and the reds. This is all scattering determined color. And I have my favorite object here called Rayleigh's fish, which is a pure Rayleigh scattering glass animal. If you shine light through it, you see the setting sun. And if you shine light on your side of it, onto it, it scatters blue. And so very nice illustration of scattering colors. But that's a white lion's mane mushroom. If any of you have eaten lion's mane mushrooms, you'll know they're pure white. And that's just put over my white light, and it's brilliant red. And we think that in mushrooms, I don't want to dwell on mushrooms, but on mushrooms, the infrared light penetrates through to the spore-producing bodies under the cap of the mushroom. So actually the cap of the mushroom is an integrating detector for detecting light, for harvesting light from the canopy of the forest and directing that light down to produce the metabolic energy necessary to produce spores. Now, I had this idea a few months ago, and I'm lucky enough to have an ex-science director of Kew Gardens as a neighbor. And I discuss everything with him all the time, but I discussed this with him. And he said, yes, absolutely. This is what must happen. But I don't think anyone's realized this yet. But mushrooms grow up like cosine correctors in the forest understory to harvest infrared light for getting extra energy for reproduction. And we believe this extra sunlight for reproduction is important for all animals, including humans, of course. The woman's skin in the abdomen gets much more transparent in pregnancy, and so that infrared light, I think, is very important for fetal development. People should listen to this. I think it's very important. So now we're getting a little bit into what we think is going on to do with the mitochondrion. Am I okay in assuming that people will know at least roughly what a mitochondrion is? It's an organelle in all our cells, in all complex cells throughout the biosphere, which has been trained to process energy and do all your bioenergetics for you. And the main engine there is the so-called electron transport chain, which lets electrons drop down a gradient of about 1.1 volts. And as it's dropping down the gradient, it pumps protons back through the membrane of the mitochondrion, where they stay until they're used to come down through the ATP enzyme, ATP synthase, which rotates and generates molecules of ATP for you. So this mitochondrial electron transport chain is basically a molecular conveyor belt that conveys food safely into usable energy. And safely is an important word there, because this electron transport can get out of control and do damage to cells. So you have to be very careful. So the electron transport chain is a controlled sequence of molecular gates that lets energy flow without chaos. You have to, you don't want everything to flow down at once. It'll sort of burst through the gate of the bottom lock and crash everything out. So you have to control the rate at which it travels down the sequence of barriers. And that's all handled by the mitochondrial control mechanisms, which are quite complex, and feedback systems and so on. You know all about this, I'm sure. I'm not an expert on those things, but so it's the cell's way of letting electrons fall downhill without losing control. They don't go downhill like somebody on a skeleton going down on ice. They go down much more gently. And I think the best analogy I can think of that is actually a flight of locks, where if you're going down a flight of locks and you're in the top pound of a lock system, in order to go down the locks, you have to fill the first lock up to exactly the same level as the water in the pound. Otherwise, you can't open the lock gate. When you open the lock gate, you can sail into the pound and then close the lock gates again, and then you can let out the water and lower yourself gently down to the next level. And of course, getting rid of that water is potential energy, and you can use that energy to pump your protons up through the barrier in the mitochondrion. So you're basically going through a flight of locks, going downhill through the complexes in the mitochondrion, and you're doing it gently because you don't want to pile up electrons together. You don't want them to suddenly drop down a big voltage gap, because they'll generate reactive oxygen species, which can do lots of damage. So you want to do all these things, you want to do gently and minimize the reactive oxygen generation. So the lock system is a relatively good analogy of that, and you don't need continuous rain to run a lock system. You can fill a reservoir at the top, and you can use that water even when it's not raining to fill the locks and run down. So I watched the Christmas lectures at the end of 2024 with Chris Van Tulliken. He builds a wonderful model for kids of the mitochondrion. And if none of you have seen that, it's worth going back and having a look. It's very, very well done. And each kid comes along carrying an oxygen atom or an oxygen molecule and runs past these pumps and bashes them on the way, and then goes and gives the oxygen molecule to the electron at the end of the electron transport chain. So this is my cartoon of the electron transport chain. So you've got the food coming in at the left. The electrons are dropping down through these series of barriers. I've drawn them not as locks, but as the red barriers. They're different heights for the different barriers you have. But when they drop through these barriers, they can push protons up through the green membrane in a membrane in the mitochondrion. And that's a huge voltage gradient across that membrane. It's very, very thin, of course, nanometer level thickness, but it's 30 million volts per meter. It's the same gradient as you'd find in a lightning flash. So mitochondria are really electrical powerhouses for producing energy. And now I'm coming back to this diagram, so I won't go into details, just to give you an idea of what we're talking about. And then there's a turbine there in purple, and then you end up with ATP molecules at the end. And this is what you use to drive that process. But I'll come back to that in a moment. So how is the electron transport controlled, do we think? Well, food provides the electrons at the top of the slope as high energy electrons. They travel down the slope through a series of barriers. The barriers are high enough to control the rate of electron flow controlled by the control system in the mitochondrion. And they need some kind of nudge to let the electron through. They can get this nudge from various sources. It can be just a thermal nudge from the temperature of your body. At 37 centigrade is enough to nudge some of these electrons through, although it'll do it very slowly. Or they can quantum tunnel through the barriers, and I'll mention that in a moment. Or they can be photon induced, and that's what I want to talk about today. You can actually stimulate this electron barrier crossing using photons. Now, the net, the, if you call this some kind of average barrier that you have to get across before you do any chemistry inside the body, that average across all of life, it's been done very extensively by studying metabolism across all life, fungi, all complex life, animals, plants, and so on. The barrier height is on average 0.66 electron volts, plus or minus about 0.05, done over a huge number of species. And that height is chosen by evolution to allow thermal noise to give you barrier crossings occasionally, slowly, but also stop it happening too quickly and give you a wheel to turn so you can actually control this through. And so that 0.66 EV is an important number to remember. So do remember it. And I say this has been determined by many studies of metabolism. And there's a wonderful book by Geoffrey West called Scale, published in 2017, I think, New York Times bestseller and so on. It's a wonderful book to read because he and a couple of ecologists who with mathematical bents tried to understand the slope of the scaling law, which they found. It was known from the 30s that there was a scaling law between the rate of metabolism in an animal and the body mass, which had a slope of about 3/4 on a log-log plot. And the 3/4 was a puzzle because if it was, just a spherical blob, it would go like geometry, it would go like surface area to volume ratio. So it would be a 2/3 ratio. But it was 3/4 to very high precision. And Geoffrey West and his people figured out why it was 3/4. And I'm not sure the biologists will believe it yet, but it was because of the way nutrients were fed through the body, through vascular systems and through the airways in the lungs and so on, which had a fractal nature. And the product of this fractal nature was a 3/4 scaling law. So I'll use that scaling law in a moment, and it gives us some understanding of what I'm just about to tell you about the amazing coincidences there are between the nature of sunlight and the requirements for biology. So just to continue with this slide, the low barrier chemistry runs wild. If we had a lower barrier, everything would happen willy-nilly and we'd end up like a blob of jelly on the floor. If it's too high, life slows down and it would eventually stop. So this 0.66 electron volts is very carefully chosen by evolution to allow metabolism to occur in a controlled manner. So in the dark, life waits for rare but statistically stable thermal nudges, and we can live at night quite happily. We survive at night. We can't survive with a high metabolic rate at night, but we generally don't want to do that. But we can survive at night. Under infrared light, this metabolic process benefits from gentle, persistent encouragement. So we're not throwing hand grenades into a cell. We're doing something very gentle. And this is what I want to try and explain in a moment. For those of you who are more interested in the mechanisms here, I've got a cartoon here of the electron transport rate, the rate at which the electron transports on the y-axis here, as a function of the barrier height here. All these barriers are between about 0 electron volts and about 0.8 volts. And so you see the kinetic energy allows you to get through the low barriers in the electron transport chain relatively easily. To get through the slightly higher ones, you need quantum tunneling. And life is very tuned for allowing quantum tunneling. If you want to get things through faster, you can move into this blue range, which is where we have the photons. And this activation energy, which I mentioned, 0.66 electron volts, is in the range where the photons are really the only things that can give you a high rate of electron transport. And that's where the solar energy peaks at 0.75 electron volts, because that's a close coincidence there. So uh, that was just to mention that the barriers, these barriers in the mitochondrion, are all about 12 angstroms thick. That's about 1 nanometer thick. And the electron coming down with an energy of roughly 1 electron volt, the de Broglie wavelength of an electron of energy of 1 electron volt is about 1 nanometer. And so that allows electron tunneling to occur relatively efficiently. So it's highly tuned to have the thickness of these barriers in the mitochondrion at the right level to allow the electron tunneling. And basically that's what keeps us alive at night, is a quantum process of electron tunneling. So how do the photons do this? In infrared light, each photon carries about 25 times the thermal energy of vibration of a molecule in your body at 37 degrees centigrade. So it's a much higher energy than the thermal energy in your body. And the nudges are derived from photons, but they don't do it directly. A photon contains much too much energy to do this process directly. It does it through distributing it through water. Basically, what the photons do, they excite vibrations in the water molecule. And those vibrations of the water molecule form a pool of quantized vibrations, which can be used by the metabolic process to bias the transport of electrons in the right direction down the hill. Now, these phonons, which are generated by an individual photon, you can generate tens to hundreds of them per photon. They go to lower and lower energy. None of them is as high in energy as the incoming photon, but they're lower energy. They decay after a very short space of time, 10 to the minus 15 to 10 to the 12 seconds, femtoseconds to picoseconds, but they're continuously being replenished. So there's always a pool of these phonons in the body ready to provide these nudges to get the electrons to move. And what this dynamic photon bath does, it reshapes the environment, the landscape around the electron. You know, you can imagine a landscape with hills and valleys and so on, which is being modulated all the time by these phonons being around, and it's lifting. It's like ripples on a pond. It's lifting the electron up and down a little bit statistically, and occasionally you'll get a statistical excess of height, and the electron will say, "Hey guys, well now we can pop over the barrier. We've been lifted up high enough." So this is a very gentle way of nudging the electron across the barrier and speeding up the rate of electrons through the electron transport chain. Incidentally, I should stress that, you know, this bath of phonons in the body when you're in sunlight can also be used for molecular repair and molecular synthesis. So it's doing all kinds of things rather than just making ATP. So when they die, these phonons, after a very short time, they're waste heat from the body. But while they're quantized photons, they're able to do useful things. They dissipate energy in a helpful way to energize or to lubricate the metabolic process. That's what we think we're done. So the process of photometabolism works as a lubricant and a source of repair, not as a source of fuel. That's a common confusion we get when we tell people what's going on. We think we're just providing extra fuel from the sun, but that's not true. Now, I've shown you that the 0.66 electron volts is very close to the peak of the sunlight, and I don't think that's an accident. I think that's a result of 3 billion years of evolution. So the chemistry that drives the height of this barrier has been evolved over a long period of time to match the sunlight as best it can. And the other amazing coincidence is that if you calculate the number of electron transport chains in your body, it's a very rough calculation because it's very, hard to do in detail, but it's around 10 to the 21 in your body. And it turns out that the flux of solar light that's entering your body is around 10 to the 21 photons per second. And I don't think that's an accident. I think that's again that's putting us squarely in a Goldilocks zone for metabolism. And so, at rest, on average, you're in the whole body, each electron transport chain is only passing about one electron every second, or even slower than that. Some can run much faster, and certainly when you're exercising, they're running much faster, up to kilohertz and so on. But basically, you're getting a rate of about one hertz for the electrons going down individual electron transport chains. They have plenty of time to wait for this oscillation of the environment, which is happening on picosecond timescales. They have plenty of time to be pushed up several sigma from statistically to be aided in getting over the barrier. So it's a gentle process, but it's persistent and it's always pushing the electrons the same way. So I just summarized there the numerical things there, the 10 to the 21 photons and the 10 to the 21 electron transport chains in the body. And that, of course, of scale with body mass in the same way. Now, just a little bit of an astronomical aside. I'm very nearly finished now, so don't panic. There's an astronomical side to this. This little peak in the solar spectrum, which I've marked in pink here, the blue line there is the solar spectrum outside the atmosphere before it's passed through the Earth's atmosphere. So you see it doesn't have any absorption lines in it, and it pretty much matches the peak of the transmitted spectrum because there's very little loss at 0.75 electron volts from the atmosphere. So that extra peak is a property of the solar atmosphere and the atmosphere of all solar-like stars, which is due to something called the H- ion. It's a hydrogen atom which has an affinity for an extra electron. The electron in the hydrogen atom can't protect the charge of the proton in the center very effectively because there's only one of them. So it can grab another electron which can hang on to that atom with an affinity of 0.75 electron volts. And this means that the H- ion is responsible for the mistiness of the solar atmosphere, for its opacity throughout the visible spectrum. And when you get to the dissociation energy of that H- ion, you reach a minimum in the opacity of the solar atmosphere. So at that wavelength, you're seeing deeper into the sun than you are at any other wavelength. And because you're looking deeper, the sun is hotter. And that gives you an extra boost above the Planck function of about 20 or 25% of light from the sun at that wavelength. And again, you see that these are the kind of energies you need to do the magic with the electron transport rate. I don't have time to explain this. This is the Marcus theory of electron transport. Marcus won the Nobel Prize in 1992 for understanding electron transport in chemistry. He's actually the oldest living Nobel laureate at 102, and I've had conversations with him over the last few weeks in Caltech because I have a friend in Caltech who put me in touch. So he has the theory that we understand how modulating the environment can change the rate of electron transport flow. So there's this coincidence with the peak of the solar spectrum, and the 0.66 electron volts here, shown in the purple here, it also peaks at around this thing. So there's this amazing coincidence that the biochemistry needs this energy, the sunlight provides it, and the life has tuned itself to work with that energy. And there seems to be enough of it. These I, Y, J, H, and K, that's astronomical mumbo jumbo for the different transmission windows of the atmosphere when you do ground-based infrared astronomy. You have to look in the gaps between the water bands if you want to do infrared astronomy from the ground. But this is coming back to the West et al. Scaling law. This is basically metabolic rate on the left against body mass on the x-axis for a whole range of animals. You can include plants and fungi in this as well, and these are all corrected for the body temperature, but you see it's an amazing correlation over more than 20 orders of magnitude. Now, we're not unfamiliar with these scaling laws because we have one in astronomy as well. This is the Hertzsprung-Russell diagram in astronomy, the color magnitude diagram, which has a main sequence here, all the main sequence stars, which is almost a power law. It has deviations, but this is astronomy. This is not biology. But basically, we're familiar with these scaling laws. And what I did while I was on holiday, actually, I said, well, okay, if we're thinking of the light having an effect on this biology, what we have to do is to show that, okay, for humans, I've just taken you through the human story. We know we have a good match between the number of photons and the energy they're carrying getting into life. What if I take a fruit fly, which is many orders of magnitude lighter in mass, and I measure the surface area of the fruit fly and calculate how much of the solar energy is impacting the surface area of the fruit fly, and assuming it's being absorbed, or some fraction of it is being absorbed by the fruit fly, and plot it on the scaling law, because I have the mass of the fruit fly and I have its metabolic rate, which has been measured. And so I plotted this in pencil while I was on holiday, and I thought I really hadn't thought it through. Had no idea where it was going to sit on this diagram. But what came out was this: that I've got a human on the upper right here with the green square, and a fruit fly on the bottom left, the green square. That's the basal metabolic rate of the fruit fly in a human. And then I've got the intercepted energy plotted in yellow there for the fruit fly and the human. And I thought, well, I won't tell you exactly what I said, but I was quite surprised. But so I realized we have this series of congruences, I call them, coincidence of congruence. There's a congruence between the matching of the energy in electron volts, and there's a congruence in the rate at which you're raining photons down on biology. And they all kind of seem to work out. It's a very weak effect. So, but because this light is not feeding energy into this line is in fact a factor of several above the basal metabolic rate. Okay, but we're not getting all that energy in the body. We're getting roughly half of that energy into the body, and we're certainly not getting half of it absorbed in the mitochondria. But we're not providing energy. We're providing just these nudges, which is what I call a lubricant. It's a lubrication that allows the process to work slightly more efficiently, but it's always working slightly more efficiently in the same direction. So that's basically, in a nutshell, what we think is going on. There's this very subtle, very gentle effect going on with sunlight inside the body, which is being used, now well trained, as being used by your metabolic process. It also turns out, I was talking to Richard Weller, the dermatologist, the other week, and we learned that the energy that you need to release nitric oxide from its storage mechanism in the body is about 0.6, 0.7 electron volts. So here we're moving the electrons, and there's another process at the same energy triggering the extra oxygen supply by dilating the blood vessels to provide, and you've got to meet these up at the end of the electron transport chain. It's the oxygen molecule and the electrons which you're meeting up. So I think there's this wonderful control mechanism going inside the body, based basically with I like to think of it as a central railway station signal box in the center of a big city, where you've got all these processes going around in railway tracks all around the city. And there's a central signal box in the center, which is using these 0.6, 0.7 electron volts to just signal which signal should open at which time and control the whole metabolic process. I think it's a very elegant, highly tuned process, which, as far as I'm aware, has just been missed. Now, we got lots of experiments need to be done to quantitatively test this. But, we're sitting here with this picture of the theory, and we've got this enormous fund of uh, experimental and clinical data showing that we are getting this effect with sunlight. I think we've got this gap where we need a detailed assessment of the theory to bring it together, but then we've, in some sense, got the whole picture. And that was my last slide. But this is a metabolic Goldilocks zone, which is much more highly tuned than the normal Goldilocks zone, which is having liquid water on the surface of the planet. This is a highly tuned, energetic and rate Goldilocks zone for running metabolism across the whole biosphere. I'm a physicist, I'm not a biologist, so I can be torn apart by biologists, but I get very excited by this for very general reasons of, the way life and the universe works. So what might be the consequences of restoring the infrared to the environment? Well, I think there will be many, but they need to be quantified more than they're quantified at the moment. The major beneficiaries may be hospitals and the NHS spending on common diseases such as type 2 diabetes and obesity. We talked about the aging of mitochondria. We talked about the effect on all the diseases of aging. And, the classical disease of aging are diabetes, obesity, you can name all, Alzheimer's and Parkinson's and so on. We know the astronauts come back with aged mitochondria, and I think this is what's going on with people who spend lots of time in built environments. Now, I was recently in an intensive care units visiting somebody, and the lighting in intensive care units is toxic. It's bright blue LED lights, no windows, not a hint of infrared, and it's toxic for these people who are living in these intensive care, not just the patients, but the nurses and doctors who are living in these intensive care units. We've now persuaded through Glenjeffrey in London, we've persuaded two of the hospitals in London to actually change the lighting in their intensive care units, particularly the neurological hospital, where they now have infrared lights in the ambulances. They can shine on neurological stroke patients straight away, because that's, I think, very beneficial. And we provided these lamps to them free of charge through a lighting engineer we have near Bath, in fact. And so I think there are many ways of improving public health. Obviously, ultra-processed food is one of them. This mechanism is not dissimilar to ultra-processed food, and I think it's at least as important, in fact, probably fundamentally more important in some sense. When on a normal diet, this becomes more important. But we are evolved to live under sunlight, and all the sunlight does useful things for us. I couldn't go through a whole range of all of them. We discussed many of them, but I think I've already gone over an hour, and I apologize for that. But I wanted to end with this cartoon. I mean, this is a freebie you might want to discuss or not, but I've extended this argument through to the universe as a whole. But somebody can ask a question about this. I'll leave this on the screen for a moment. Thank you.
Thank you, Robert. That's wonderful. I'm wonderful. Thank you so much. Some questions to digest already, but yes, I mean, so much is so interesting there, and it's the attunement aspect to it. I think is going to be very natural to this audience of cybernetics here, in the way in which these things.
I thought it might, yes.
Yeah, fit together, but that's going to be maybe part of the discussion. Alexander left with a question. He had to go at the top of the hours, but he left with a question, and the question he left was basically the expected question, which you anticipated, which is, what about all the animals that live deep down in the water?
Yeah, I mean, these are very interesting and important questions, and we don't know all the answers. But basically, animals that live in the dark, we do know this, that live in. There's a salamander that lives completely in dark caves and only gets food from photosynthesis in the surface. They metabolize extremely slowly. And the salamander, which is really quite a small animal, lives for 100 years. So if you go back to these scaling laws, which are developed by Geoffrey West and so on, you can fold in the lifespan and the rate of aging and the rate of necessity to repair your body, and this all kinds of comes out of these scaling laws. So the animals that live deep live slowly. The animals that are very big live slowly because your metabolic rate, obviously per unit mass, decreases as you increase in size. So you can pick examples of animals that live in unusual circumstances, like in the dark. And I think you can relatively easily explain how they fit into this overall picture when you realize that it's to do with metabolic rate, it's to do with not doing damage through generating too many reactive oxygen species and so on, and living at different rates. So it's not an insoluble problem. Glenn Jeffrey and I have been discussing the idea of animals that re-enter the sea, the hippopotamus and so on, that became the whales. Why did they go back into the sea? What was the advantage to them of going back into the sea? You can see the advantage for animals to leave the sea, because as soon as they reach the land, they get the benefit of all this infrared radiation, which they would only get intermittently when they're very close to the surface, when they're in the sea. So as soon as animals and plants came out onto the land, a huge niche space open to them, which wasn't available to them before. And this is because their metabolism is so much more effective with the infrared radiation. So we really need these extra nudges, and without them, we have to rely on quantum tunneling, which I've said is perfectly adequate for keeping you alive in the dark. But your opportunities, I mean, think of the lizards, the reptiles that come out and sun themselves on a rock in the morning. Any biologist will tell you they're cold-blooded animals, they have to warm their body up. Sure, don't say that's wrong at all, but they're actually turbocharging their mitochondria so they can predate or avoid predation. And there are easy experiments that could be done. We're not licensed to do experiments on reptiles and so on, so we can't do them ourselves, but we're hoping that other people are going to look at reptiles and put them out under bright lights containing no infrared and bright lights containing infrared, and see which ones that you can think of all the experiments you could do on reptiles probably very easily without doing them any damage. So yes, it is an important question, but we are very sensitive to it. We're thinking about it, and it opens up many exciting possibilities of weird animals like bats, for instance. Bats have extraordinary abilities at repairing DNA. They're full of viruses, but they spend a small amount of their time in daylight, dusk light, and they have these enormous surface areas for collecting infrared light. So they can probably get enough infrared light in those periods in order to give them their high metabolic rate. So yes, all those are interesting questions. Do you want me to oil the process by going through my diagram here? Because
Yeah, of course. Yeah, we should come to that, and maybe also some of the public health implications as well. But let's have a look at the diagram first. Thank you.
Well, the diagram is basically this idea that there are three things that are important for generating complexity in the universe. One is a surface which is cold, one is a source of energy, and one is an entropy gradient. So if you think of a star forming in the universe, the sky around the star is cold. All the material in the interstellar medium is coming down, and there's a gravitational pull on a extra dense region of nebula, and you form the star. As you form the star, as you collapse it, it gets hotter because of the bicycle pump effect, just thermodynamics. And then if it has enough mass to ignite nuclear fusion, you trigger an energy source in the center. And that energy source in the center enables the star to maintain a stable equilibrium, quasi-stable equilibrium for the lifetime of the star, which in the case of the sun is about 10 billion years. And there's a delicate balance between the energy being produced in the core of the star by nuclear fusion and the entropy gradient in the material all the way down from the surface of the star, which is controlling this the configuration, the low entropy configuration of the nuclear burning region in the center of the star. And all the extra entropy that you're generating in this process is radiated as starlight. It takes it a while to get to the surface of the star. So you have an energy source in the core of the star, which is nuclear fusion. You have a source of waste entropy, which you have to radiate into the cold of outer space by radiating from the surface of the star. While you're doing that, you're generating the periodic table by nuclear fusion processes in the core of the star. So you're generating the periodic table as you evolve the star. And you have this cycle of radiating to a cold surface, generating energy and utilizing this entropy gradient to maintain the stability. When you get a star and a planet, the star is radiating the surface of your planet with its waste entropy, which is in the form of low entropy photons in the solar spectrum. Those photons are absorbed by plants and animals and so on. They're producing life. You're maintaining homeostasis, which is like the stability of the star over 10 billion years, except your lifetime's a bit shorter, but you're maintaining homeostasis from this energy coming in from the sun. And you're generating biological complexity. You're evolving and you're generating biological complexity. So you have two processes which are very, very similar. The star in a cold universe, the planet in a cold universe with a star nearby, which is going through the same process. Now, whether you call one alive and the other not, I don't know. That's a debatable point, but they're very, very similar processes as far as physical processes are concerned. And I find this extraordinary. Then we can get onto black holes and Hawking physics, which I'm not going to do today. But that is a
Next
a
month.
natural extension of this in a way. So there are these very general principles in the way the world works, and life is so closely coupled to these principles. I'm very now, having gone through all these arguments, I'm very convinced that many Earth-like planets in the universe will have carbon-based life. We can even predict from the mass of the star what kind of life there might be. I won't,
I see
unless
that.
you ask me the question, I won't tell you why. But I think there are some general principles here which we can begin to understand in terms of astrobiology and the kind of life you might get. It's a bit speculative, but it's always fun speculating. So. Anyway, I've done enough talking.
I mean, that's wonderful. If I can take the next question as well, obviously, anyone else who wants to come in, please interrupt me. But because we did touch on the kind of very practical public health implications here. I mean, the figure from Saudi Arabia is quite stark. An interesting contrast because the amount of sunlight actually available in the country.
They never go outside. I don't think they don't go outside at all. They go from their car to their skyscraper, and it's all air conditioned.
But then there's the question of say a country in the northern hemisphere like the UK, for example. It's a good time to take this talk, Bob, because, as you say, you haven't seen much sun for a while now, and we're living inside with these LEDs as our new companions, and we don't have infrared. We don't have even our old style bulbs. We don't have. We don't read under candlelight. This kind of
I do.
thing.
60 watt bulb run at half voltage.
Yeah, interesting. You do, yeah. Okay. I think a few of us might soon. um, so this is, I mean, it, how much evidence is there on this part of it? Are we seeing diabetes rates, for example, and can we correlate them in some sensible way to housing types, for example, as you did with the Saudi example?
Why don't you talk about Glenn's work on that, the measurements and
Yes,
so on?
yes. I mean, yes, there is a lot of work that's being done on this. Glenn and Mike Powner, his colleague, did an experiment a year or so ago where they looked at controlling blood sugars using infrared light. So they used themselves as guinea pigs, and they starved themselves with glucose for some period of time, and then took a huge glucose dose, which is very unpleasant, and then monitored their blood sugars for 24 hours afterwards. And there were two experiments, one with red light shone on their body, actually an LED, an 850 nanometer LED on the body for 20 minutes when they took the glucose, and a dramatic difference in the behavior of the blood sugars, much lower peaks with the infrared radiation. And also you could see the increased carbon dioxide because of the more rapid metabolism. I have an anecdotal example with my wife who went on one of these Zoe tests, and she took had one of these sugar monitors on her arm for a fortnight, and did all the sugar challenge, the glucose challenge tests, and so on.
Yeah.
And she got her results back from the Zoe people, there was one thing there which was way off what was expected. The response for a woman of her age and sex, obviously, was something like 40 on the scale they were using. I can't remember what it was, but her result was something like 87, and it was many sigma away from the expected rate. And we've had an infrared lamp on in our bedroom every morning. For 20 minutes before we get up. We'd had it on for a year or so. So her blood sugars were extremely well controlled during this stress test. And I had a similar example. I went for my annual blood test with my GP, and I walked into the surgery when I was invited back to discuss the results. And as I walked in the door, she said, "You don't have type 2 diabetes." I didn't think I had, but that's the first thing she said when I walked into the door. So, there is experimental evidence. There's also experimental evidence that Glenn and people are doing at the moment, that for people who live in windowless offices, if you give them an ordinary tungsten filament lamp in an angle poise like this in the big office there, and you give them one each and test their visual performance, which is a very good mitochondrial function test, test their visual performance before and after, there's a very significant improvement in visual color contrast tests. And it's better for tungsten filament lamps than it is for LED lamps, which have a restricted wavelength range. And that's consistent with our view that it's the whole range of the solar spectrum which is doing the work in metabolism. It's not a single process, it's multiple processes, and all of the solar energy is being used in some way or another. And we've evolved to have that happen.
Yeah.
So if you're in these extremely artificial environments, like a windowless room with white LEDs and nothing else, you're in trouble. And in these rooms in London, these people were off sick all the time. They were turning up late for work. They had headaches. They didn't want to work there. And when they took the tungsten filament bulbs away from them, they all screamed, said, "No, no, 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 we want to keep them."
Interesting.
And yeah.
So, should we take Mauricio?
Yeah, please.
Greetings to all. Thank you, Professor Robert, for a wonderful talk. I have zillions of questions, but I'll reduce to two. questions.
Yeah.
One is punctual. What is in that graph that you showed before? I don't remember the slide where you defined the regions, where the traditional way of lighting seeds, so incandescent bulbs.
Yes.
They should have more infrared radiation than the modern the modern lighting that we have now.
I think for the seeds, we know that longer wavelength red light, far red light, has a major effect on plant growth and so on. Again, I don't know all those details myself, although you've got an easy way for me to find out. So this is the region where both plants and animals are very transparent, and it's around 800 nanometers and 1 micron, 1.2 microns. So you know I've done a number of experiments with my integrating sphere where I look at the basically the back reflectance of plants, living tissue, other living tissues, humans, seeds, fruits, and so on. They're all extremely translucent in this range where the metabolism is occurring. So I think it's it's relevant to the mitochondria and the plants and how the mitochondria control what plants are going on.
Mm.
The second question is, well, cells do adapt to meso conditions in photons. So they have different metabolism pathways when you have real light and when you have low light or no light.
Yes.
Why we shouldn't be searching this in higher organisms?
Why? Sorry, could you say it again?
Why doesn't have the higher organisms, the more complex organisms? Why is there not in their metabolisms different pathways to adapt to low conditions of or to different conditions of lighting?
Well, I think we do adapt. I think we adapt with circadian signals. Certainly, if we live in normal lighting, we adapt to circadian signals. We're doing different things at different times of day. We also know that the irradiation of mitochondria with this near-infrared light is much more effective in the morning than it is in the evening.
That's right.
And that's because your mitochondria are becoming very active then. So yes, I think. I mean, I wish I understood more about the control mechanisms in the mitochondria, but there are these exquisitely sensitive control systems that can control the rate at which things happen and control the other pathways that are necessary to link into just the basic electron flow. So I think it's a wonderfully complex system, but
what's also interesting, Rob, on that is the intensity of light matters for our biology. And you can see some of the epidemiological data from the UK Biobank studies.
Yes.
I think the one paper was called Bright Nights, Dark Days.
Yes.
Which showed, something like 30% increased probability of chronic ill health, mental ill health, etc. If you had insufficiency of light intensity during the day, which is really quite useful to use a light meter on your phone to measure your lux. It's a simple way of doing it during the day, because it turns out we need at least 10,000 lux to sort of get good cortisol action in the morning, get everything pumping up, hormone signaling, etc. And then not just the chromophore activations, etc., but then at night, if you've got excessive brightness at night, and especially in the blue wavelength, apparently, this has massive knock-on effects too. So then the recommendations there are to make sure you've cut your light intensity significantly and your bandwidths by 9 o'clock at night.
Yes. Yeah, I mean, this is sorry, this is this is all to do with signaling, isn't it? If you're signal your body to do something which you can't do at that
Correct.
time,
Yeah.
because it say doesn't have sunlight, for instance, you're in real trouble.
That's right. And if the intensity isn't enough, you have to wait a long time to get to the right threshold to be able to activate the signal properly. Yeah.
But it's not just the intensity that's involved here. I think it's the ratios between lights. You know, we know that blue light on its own is very toxic. But blue light with the whole solar spectrum is absolutely fine because we've lived under it for, you know, hundreds of thousands of years. So, you know, there are ratios. And I mentioned about the dynamic range and brightness of the light. There's a dynamic range in the triggering of these processes as well. And experiments that Glenn and people are doing in London showing that the triggers of the infrared light on metabolic process can be very, very low, much lower than sunlight.
Yeah.
But it's, you know, it's other factors in the environment, like the ratios and so on, are so important there. So it is a very complex system. And, you know, I'm talking to the right audience, complex systems, but you realize that, you know, there are some basic fundamental physical effects that are going on.
Yes.
But those physical effects are being integrated with very complex control systems.
That's right.
And it's you know, it's going to be a nightmare to tease
to try and figure it all out.
Although I think, you know, my feeling is, I mean, I spent 50 years or so doing astrophysics, and you know, this is a second career for me. But you know, I've learned a lot about biology and some about chemistry and so on in doing this. But I'm certainly not an expert in those fields. But I think it's made me realize that there's this very special interaction between physics and biology at the moment, which is absolutely ripe.
It's magical, actually. It's lovely.
And I think, you know, astrobiology has some role to play in this because, you know, astrobiology has been extremely popular over the last, and it's well funded now by the European Union and so on. So, and this astrobiology, I mean, I talked about predicting what kind of life you get on different planets and so on. There are ways that we can see that you know, we get levers on great mysteries in the universe by studying these things. So if I were going to start my career again, I think it would probably be in the interface between life sciences and physics. I think it's tremendously productive. And I think this book by West.
Yeah, it's a great book. Yeah.
And you know, the biologist, so I have to quote you this little bit from Roger Penrose's foreword to Schrodinger's book. If you've never read the Roger Penrose foreword to Schrodinger's book, it's only a page long, and I thoroughly recommend you read it. It said, like so many works that have had great impact on human thinking, it makes points that once they're grasped have a ring of almost self-evident truth. They're all still blindly ignored by a disconcertingly large proportion of people who should know better.
Yeah.
How often do we still hear that quantum effects can have little relevance in the study of biology, or even that we eat food in order to gain energy? This serves to emphasize the continuing relevance of that Schrodinger's "What is Life" has for us today. It is amply worth rereading.
Yeah,
I've reread it 5 times.
Wow.
And it's an amazing collection of wisdom. Certainly about entropy and basic physics. It's a wonderful story.
I think we have Elena and then Alan with hands up. And also, Peter, we could turn off the recording, I guess, now. But Elena.
Thank you for a very interesting talk. I wanted to think a little bit more about the public health implications. Have studies been done on people who work night shift? And if they do work night shift, does it matter if they sleep in a room that has natural light through a window?
I think there have been studies done on people. I mean, people who do night shifts are generally not very well. I think they suffer quite severely from this. I think. Sorry, can you remind me the first part of your question again?
I basically just wondered about the health effect of working night shift,
Yes.
And if it was somewhat mitigated if they slept in a room during the day that had some natural light through a window?
Well, I think probably during the day they would certainly want to be in. They would want to have access to some daylight,
Infrared.
infrared and daylight. But on night shift, I think if they were living under more natural lighting with an infrared component, they'd probably suffer less.
So,
but, you know,
and Bob, on that part, Moore Eid has done some work on that. And I could just point people to his research on that, on the circadian effects. And he's actually now manufactured a bulb which removes the 420 to 450 nanometer wavelength.
Yeah.
And he's found that circadian effects seem to dissolve, as long as, but he's not looked at infrared, seem to dissolve as long as he takes that wavelength out. People have less circadian disruption working night shift, but then they need to be in the dark during the day. So they're going to be deficient anyway on both ends, right?
But I think for all of these environments where people are working night shifts, they need to take the first account of this.
So you have to have the
having an extended spectrum.
Yeah, you have to have the darkness, you have to have the full spectrum. Yeah.
I think I'd make a general point about artificial lighting. I mean, we could talk forever about artificial lighting. First of all, I say we are in very, very close contact with the lighting industry. We're talking to a lot of lighting industry people. We have many joint talks and seminars and so on. We're also talking to architects. We're talking to Norman Foster and Partners and have been for quite some while. They're very concerned with this because now they know that they're producing toxic environments. They have to take it very seriously indeed. So there is a lot of work being done on this. But I think the point I would make, we have a lot of trouble with regulation of tungsten filament bulbs. They're illegal in the US. They're illegal in Europe. I'm not sure they're illegal here, but they're hard to get. But the problem with tungsten filament light bulbs is they're very inefficient producing visible light. That's their problem. They use lots of energy to produce infrared light. So we don't need tungsten filament lamps to produce visible light. So I run all mine at low voltage. So they produce a bit of visible light, not very much. But the filaments last forever. And so they're 100% efficient at producing infrared light. And infrared LEDs are very, very inefficient, in fact. So these are the obvious things to use for the kind of effects that we've been looking at. But the thing is, we only need these lights to follow the bodies, the people who are, or the animals, or the whatever's alive need the light. You don't need it for illuminating stairwells and toilets and all the other places. So you just need to carry this light around with the people, and it's then relatively straightforward, and it's cheap because you don't take this out of the heating budget, the lighting budget, you take it out of the heating budget because all the light that's gone through your body comes out as heat anyway. So you're heating the building with the light. You should always heat with a temperature of above that of a candle flame. It's a waste of high quality electrical energy to heat a radiator to 50 degrees centigrade. You need to heat something to the temperature of a candle flame and shine it through somebody first, and you still get the heat out of it. So it's an organizational problem rather than a technical or funding problem. But these different environments, like the shift environment, I think people do suffer terribly in shift environments, and you could swap the complete darkness in daytime with an infrared rich environment, not a red rich, an infrared rich. The trouble with the red LEDs is they're red, and it's very uncomfortable to work in those. But a lighting with an infrared component is perfectly natural to work under.
Alan has his hand up.
Alan, thank you.
Thank you. Yeah, thank you, Bob. That was fascinating. All of my questions are very short, and they all begin with the letter B. Basically, bacteria. Does that have an influence here because it doesn't have mitochondria, and I guess viruses as well? The brain, because I know you showed that wonderful image of the bamboo, and without the skull, which protects the brain, it allows light through, I assume. So maybe that's a benefit. My third B is bark, the bark of a tree. How does that think about maybe evolutionary, how has that benefited from this process? Because all the living cells of a tree are directly underneath the bark.
Yes. Well, first of all, bacteria. There are experiments on bacteria, many of them, and they reproduce many bacteria. I don't think all of them, but they reproduce much more quickly under infrared light. And I know that worms, for instance, produce worm casts much more quickly under infrared light, factors of 2 or 3. It's actually used for product worm cast production. The brain, yes, light can get deep into the center of the brain. There are lots of experiments on Parkinson's disease on the brain, and we know it gets in. There are lots of neonatal experiments are done where you can look at neonatal health by shining an infrared beam through the skull. As I showed in the pictures of my hand, the bone is perfectly translucent in the infrared. It doesn't stop the light getting through. Bark, yes, well, bark is very transparent in the infrared. I mean, I do experiments on living tissue. You've seen the wood. Bark as well is very transparent, and
interesting.
Yeah.
So what's also fascinating, if I can sort of pop in on that, is how there are the difference. If you mentioned the LEDs and the sharp spikes, and you look at mitochondria, they know the difference between an LED source of red and infrared and the type of lamp you've got, Bob, on your desk there. That sort of Philips bulb. Do you want to talk more about that? This actual shape of it.
That's also very interesting. That's another problem we've looked at quite recently, and it's very. If you use these intermediate band LEDs, like an 850, it has a bandwidth of a couple of tens of nanometers. So it's not like a laser, but it's relatively modest bandwidth. If you generate as much energy in that LED as sunlight, I think you will tend to saturate. Sunlight, if you expose a body to the full spectrum of sunlight, it's very hard to saturate these processes. The process has a sort of biphasic response.
Yeah.
It's not a normal dose response. So the more light, the more response you get. It's a switch. Once you switch it on, it works. And if you go too far, you can switch it off again. So it's a biphasic process. And I think we have to be aware of that. And I think, the LEDs, a lot of people want to make multiple LED systems that provide an approximation to daylight. And they do that very successfully. I work with one called Steve Paolini in Silicon Valley, who does this with multiple LEDs. And he can dial up different spectra for different kinds of daylight and so on easily with a simple algorithm. But it's not providing the infrared. And basically you need the infrared. And so all these narrowband LEDs, which will interact with maybe a particular chromophore, they won't necessarily react with water unless you put them in a water band.
But even the LED, the red and infrared LEDs, because I've got an LED lamp here, and I can feel the difference, by the way, in my body, because I'm not well. So I can actually feel the difference between an LED infrared and when I'm outdoors infrared. It's a completely different feeling in the body.
And Glenn's experiment showed that the LED works, but not as well as a broad spectrum lamp. I just come back to evolution again and say, we have evolved under sunlight, and all of our systems have reacted to that driver over many a very long period of time. And as soon as you mess with that, you mess with the ratios, or you mess with putting too much intensity at one particular wavelength, I think you're going to get in trouble. So
I think it's a real lesson for cybernetics, which I think we can perhaps engage in over coming conversations. of the restriction of variety that's been removed by our actions of moving indoors, and then the light situation of optimizing one segment, but of course diminishing the others by so focusing on efficiency and not health effect. So yeah.
Well said. Peter, thank you. I know that there's an interesting, important comment by John, just attesting to his own experience in the comments. But also, as Farid had raised one or two points earlier, we missed him, I think. But Farid raised the question of the octopus, which I think perhaps you feel you've dealt with already, Robert. But is there anything we should be saying about any particular kind of undersea existence or species? Are there differences, or should we just refer back to your earlier answer? Because
I think the octopus is very, very interesting. And I'm very interested in the octopus as well, because they have enormously short lifetimes, don't they? 1 or 2 years. And I think, and of course, they have a different kind of blood, different reaction. But I think I'd love to study the octopus. I can't answer your question, and I can't explain the octopus for you off the cuff. But I think it's one of those outliers which can be a very good illustration of what actually is going on behind the machinery here.
Yeah, interesting.
It's an extraordinary animal.
Interesting question, Farid. Thank you. Good. Is that us? I know that you're due to be delivered back to your wife by 7.
Yes.
I mean, that's been extraordinarily powerful, Robert. I mean, thank you so much.
Thank you very much.
Yeah, I mean, absolutely. I mean, philosophically as well, as Peter was just saying, you know, about how ultimately the cybernetics manifests at different levels of recursion. So much to think about there.
I'd just like to make a plug for interdisciplinarity, because
absolutely.
I stopped being an astronomer, and I didn't stop being an astronomer, but I did look at other things, and I've had a very interesting experience of talking to people in completely different fields like biology, botany, at a high level, because you don't have to climb all the way up these other fields. You can branch over at the cutting edge and interact very effectively, because you offer this entirely different perspective of the same problem, and that's incredibly productive. And I think it's in a way it's a wasted resource. I mean, I had to retire from the European Space Agency nominally at 60. I made it till 62, but then they kick you out, and I thought,
Well, just it is a tragedy, and I think cybernetics particularly might have something to offer in that interdisciplinarity, of providing a language to connect up and to examine even the consequences of those decisions which are taken in isolation, and then have the monumental effects they have on our health and the health of the planet and the health of our brains. I mean, we're doing it to ourselves through ignorance, and I think your work is just wonderful and commendable, and I hope this society can support you in whatever way you would wish, in so many ways, and I'm certainly open to working with you in that regard. So thank you.
Could I make just one comment there? I think one thing we need is to spread the word, and we do lots of podcasts and so on. But at the moment, the government and the nutrition industry is completely blind to what we do.
Are.
And I listen to these nutritionists, and I listened to a story about forest bathing in Japan the other day. You may have heard it, where they talked about all these terpenes and so on interacting with your body. Yeah, they're fairly strong stuff, I think. But nobody mentioned the fact that the understory is incredibly rich in infrared.
Yeah. I mean,
I've seen it written in papers now that there's no infrared in the forest understory. I mean, papers say there's no infrared at all. It's complete bullshit. It's.
I love it. I love your mushroom analogy too. That was wonderful.
know we've got John with your hand up.
John, and
we
a couple of
have
minutes before we've got to call this, and I'll stop the recording on the hour.
We've got about 2 minutes, John, but that was
I just wanted to throw in another little anecdotal contribution. About 25 years ago, I had the good fortune to spend about 2 years living right in the middle of mid Wales, in a very dark valley. It was probably the happiest time of my life, because at night, on cloud-free nights, I could see full range of stars. Neighbors would come round. We'd just spend time round a natural fire, telling each other stories, looking up at the stars. And if I went to neighboring valleys, I could actually completely block out all unnatural life. It was wonderful. Since then, I've been completely spectrally abusing myself.
Yeah.
So fortunately, Margaret has come to my rescue and
absolutely
educated me a bit. So you can't see it here, but I am sitting in a red shifted light, and in the daytime I do use a lizard lamp to try to adjust my other parts of my spectrum. It's not perfect, but I notice the effect.
I've got one of those as well, the reptile lamp.
Excellent.
Yeah. May I just make a comment about the brain, because that came up as a question. Bob, I don't know if you're familiar with the work, the 1064 nanometer range for dementia or cognitive decline, to help reverse it. They've designed helmets at University of Durham.
Oh, you mentioned that. Yeah, you mentioned
that.
Yeah.
I can't remember the chap's name. And so anyway, they've actually started trials with this. And they got special LEDs made at nanometer 1064. And even Terry Pratchett, by the way, before he passed, tried the treatment to try and reverse his dementia. And they reckon what it's doing is it's actually structuring the water in the brain tissue, which is then allowing the tau tangles and so on through the extracellular vesicles to actually just pull that out. So that's quite interesting, I think. I've not followed up on that for a few years, but
in a sense, this is our phone on bath. We are
there we go.
Water as well, right? I think water is the main chromophore, we think, in this. And there are always stories of reducing viscosity and so on,
That's right. And I think also for folk who've got autoimmune conditions, it's extremely important, that phone on bath.
I think we should probably let Bob go.
Thank you, Bob. That was wonderful.
You've got 45 seconds left, actually. Anyway, it's been a delight to talk to you, and a delight to hear the interest that you have. And
No, thank you. That was wonderful.